Welcome, everyone. My name is Louise Smoke. I'm the Programs Director for Friendship Heights Neighbors Network. I'd like to spend a couple minutes talking about villages. This program is organized by a village, Friendship Heights Neighbors Network, in collaboration with Little Falls Village. Both FHNN and Little Falls Village are small local nonprofits who exist to assist neighbors who wish to age in place. You may already be a member of a village, but if you're not, I suggest you look into villages. Perhaps you might want to become a member. Perhaps you might want to volunteer. I certainly have enjoyed my association with FHNN. And now it gives me great pleasure to introduce Dr. David Rabin, who is a board member at Friendship Heights Neighbors Network.
Oh, thank you very much, Louise. Our speaker today, Dr. Suzanne Epstein, has a BA degree from Harvard in chemistry, followed by another several years in Boston, getting a PhD in biology at MIT. She then spent several years at NIH and joined subsequently FDA, the Food and Drug Administration, where she worked for some 38 years, mainly in the Center for Biologic Evaluation and Research. As a principal investigator there, she directed a large research lab in immunology, which involved training and mentoring staff, students, and postdoctoral students, designing their projects and publishing their results. Her lab focused particularly on universal influenza vaccines. She also did regulatory review of applications to FDA, investigational products, and to help develop regulatory policy, and did some research management. She just retired recently, and I, for one, much admire her willingness and interest in sharing her knowledge of viruses to today's audience. So thank you very much. Look forward to hearing you talk, Suzanne.
Thank you. Good afternoon, everyone. Thank you for coming. And I want to thank David Rabin for this opportunity and Louise Smoke for the coordinating of the meeting. I'll be talking about immune responses to viruses and vaccines. And first, a couple of disclaimers. This talk represents my views. It does not represent the FDA officially, where I worked before retirement. And the talk is intended to provide an understanding of immunity to viruses. It does not give specific advice about what any individual should do. Infectious diseases caused by viruses are a major public health problem, as we know. And I'll try to cover a few main points supported by other information. There are multiple types of immune responses that result from either infection or vaccination and play roles in protection. Immunity can prevent illness or make it less severe when infection does occur. And viruses evolve over time. Sometimes they escape our immunity, calling for a booster or a new vaccine. So here's an outline of what I'll cover. First, just one slide, a little information about the history of vaccination. Then I'll describe how the immune system works, then viruses and how they change, and then I'll use two respiratory viral diseases as case studies, influenza and COVID-19. Even before there were any vaccines, it was long known that people who recovered from certain diseases couldn't get them again. It just wasn't understood how that worked. And before the first vaccines were developed, there was an ancient practice called variolation. In that, material was taken from a smallpox lesion and administered to a healthy person to try to prevent smallpox. It was preferably taken from a mild smallpox case. And it was somewhat effective, but somewhat risky. Then in the late 1700s, the smallpox vaccine was developed from a related but milder virus. And then in the late 1800s, the rabies vaccine. Starting in the 1930s, vaccines for influenza, polio, measles, mumps, rubella, and more recently COVID-19 and RSV were developed, and there are others as well, hepatitis and so on. The map shows regions of the world where smallpox was eradicated and the dates at which that occurred for each region. And there's a timeline of these different eradications. For example, the North America in the 1950s. In 1980, the World Health Organization declared smallpox eradicated from the whole world. Now, the immune system was completely unknown in the early part of this history. It was not at all clear why these effects happened. So turning now to the immune system and how it works. There are three main types of responses I'll cover: non-specific ones, antibody responses, and T-cell responses. Non-specific responses are also called innate immunity, and they're not specific for any particular invading disease organism. A disease organism is called a pathogen. These non-specific responses can be triggered by a wide range of different pathogens, or even by an injury with no pathogen present. They provide an immediate defense because they begin within hours, and these include responses like antiviral proteins of various kinds, white blood cells that flood into the area, and an inflammatory response. These are all short-term, but they can enhance the later specific immune responses. Turning now to the specific immune responses, the first one I'll describe as antibodies. And antibodies are proteins in the blood and secretions, for example, in the respiratory tract or the gut and other parts of the body, that recognize specific pathogens and help eliminate them. These are made by B lymphocytes or B cells, a type of white blood cell. These cells secrete antibodies. And antibodies are a protein complex. They have two kinds of chains. Shown in green is the longer one, the heavy chain. Shown in yellow is the shorter one, the light chain. And this complex has two of each. This is a certain kind of antibody unit. And there are other classes of antibodies that have multiple units like this. Now, how do antibodies recognize a target? They use a lock and key recognition system. That means they have binding sites which fit the target molecules specifically. The targets, which are termed antigens, are molecules on the foreign invaders, such as viruses. So here's a cartoon antibody, and there's a curved binding site on this particular one, so it can bind to this curved antigen. Here's one with a pointy binding site, so it can bind to this pointy antigen. And this is, of course, just diagrammatic. It can be, in 3D, a protein structure. The tighter the fit, the more effective the antibody will be. And an antibody that binds to the outside of a virus, to one of the outer proteins, can block infection. It can prevent the virus from even getting in because the antibody's in the way. And that's called neutralization. It can really prevent infection, but not all immune responses do that. Now, how is it possible that we can make specific antibodies to so many different targets? It includes other things as well, bacteria, fungi, but it includes viruses that have never existed before when new viruses emerge. How can we possibly do that? Well, shown here, the antibody now is shown differently. The blue portions on both the heavy chain and the light chain are the constant regions. Those are the same in all the antibody molecules of a class, and they carry out important functions that are not specific to a certain target. That's things like coordinating with other cells in the immune system. But the red squiggly parts of the heavy chain and the light chain are the variable regions. These are different in each antibody, and they bind to a specific target. This region up here has the combining site or the binding site. the receptor for the target. So they can bind to a specific pathogen. We have multiple genes for the heavy chain variable region, multiple genes for the light chain variable region, and then combining different chains. So we end up with a huge repertoire of combinations. Cross reactions are important with viral variants, so I'm going to mention them. A cross reaction is recognition of different target molecules that are similar in some way. So here's our curved antibody. It can bind to this as well as to this surface here, even though other parts of those molecules are different. And then this one is still sort of curved, but the diagram is intended as kind of squashed to say it's not identical, but it can still be recognized. It's somewhat similar. So a perfect match is not always necessary, and that's important for vaccines. Here's another case where these two targets have pointed portions, but differ otherwise. How long can an antibody response last? For different responses, anywhere from a few months to lifelong. Some durable ones, long-lasting ones, include measles, mumps, rubella, and smallpox. But unfortunately, the responses fade much more quickly to some other viruses and vaccines, including influenza and COVID. Turning now to the other kind of specific immune response, this is T-cell responses. T cells are certain white blood cells that recognize foreign targets bound to the surface of cells of our bodies. Some of these T cells patrol the body to detect and kill infected cells. They're called killer T cells. That's in order to prevent the infection from spreading further. Some enhance other immune responses, and they're called helper T cells. And then there are some T cells that control other immune responses so they don't go wild and cause damage. Those are called regulatory T cells that can put on the brakes. So here's a T cell, and it has a receptor. The little red V is a viral peptide. That's a piece of a viral protein, and it's being displayed on the surface of a host cell. That's a cell of our body that's infected. And you can see the little black square is a host protein, one of our proteins that is in complex with that piece of virus. So the T cell receptor, this protein on the surface that recognizes a target, in certain ways, it's like antibody. These receptors have variable and constant regions and multiple chains. And just like antibodies, there's a large repertoire. But there's a difference that's important. T cells recognize their target, the piece of virus, only on the surface of a cell in combination with a host protein. If the viral protein is floating free in solution, T cells will not recognize it, but antibodies would. Now, what is immune memory? It means that prior exposure leads to an enhanced response when a target is encountered again. Both antibodies and T cells have this, and if the body has encountered the same target before, the immune response rises more rapidly and to a higher level. So here's an example. This is antibody in the vertical, and after the first or primary exposure, there's a lag, a delay before anything happens. Then antibody rises gradually to a modest peak and then falls not quite to zero, but to a low level. If there's a secondary exposure, and either of these could be a vaccine or either of these could be an infection, they could both be infections. This time, the delay is shorter, the response rises more rapidly and to a higher peak, and then falls to a higher plateau. So it means that after one exposure, and that can be a vaccine or an infection, the body's in a state of readiness with potential to better control virus and prevent illness. Immunity can protect different sites in the body. The antibodies and T cells are not only in the bloodstream, they can be at the site of infection, so they're available in the right place, and that can be very effective. For example, immunity in the respiratory tract from an intranasal exposure by infection or by vaccination. Some viruses enter through the air, And then the cartoon is an intranasal vaccination, for example, FluMist, which is given by a nasal spray and immunizes the respiratory tract. Well, does any of this actually protect us? So here's one example. This is acute hepatitis B in children. We're looking at the rate of hepatitis over a period of years and the percent vaccination. So this plot is the rate of hepatitis, and it was high. Then at the time point of the arrow, universal infant vaccination was recommended. And here's the vaccine coverage. It starts to rise. And as it rises, the rate of disease falls. The benefits of immunity, include immediate defense against disease and injury, clearing of an existing infection when one gets started, and then preventing repeat infection, or at least making it milder. Now, what's the evidence as to which response is really doing the protection? Here's a case with antibody. We're looking at respiratory diseases, and antibody deficient people have a condition where they don't make a normal amount of antibodies, or in some cases, any at all. And we're comparing to normal controls. So looking at their rate of respiratory disease over time, there were a lot more respiratory illnesses in the people who lacked antibodies than in the normals. Well, what about T cells? In AIDS, which is caused by the virus HIV, there's a T cell deficiency that progresses. As the T cell numbers decline, the patients are prone to more and more infectious diseases. So here's their count of, a certain kind of T cells over time. And as their T cell count drops, they get more and more infections. And I'll just mention that people who have a serious deficiency in both antibodies and T cells are extremely susceptible to infection. That's a very serious condition. So immunity is a balancing act. This is true whether it's due to an infection or to a vaccination. It has the benefits of clearing infection, protecting against reinfection, and some tissue repair after recovery. But there can be harm, inflammation, some tissue damage, and then autoimmunity, which can be a serious disease. Well, why did we develop harmful responses in evolution? There's some interesting reasons that we can discuss in the Q&A session if anyone's interested. Then vaccines indeed can cause some side effects, which are usually mild, not always. But keep in mind that harm is much more likely from the disease caused by a virus than caused by immunity to it. So here's an example of that. Influenza vaccine can cause rare cases of something called Guillain-Barré syndrome. That's a form of muscle weakness. It happened mainly with the 1976 swine flu vaccine. So that's a concern and is always watched for. But influenza itself causes a lot more of Guillain-Barré syndrome, about 17 times more. And getting the flu is more likely without vaccination. If anyone's interested in safety testing or the vaccine adverse events reporting system, we can discuss that in the Q&A session. Immune response is very individually. Each of us has different levels. And factors that matter, age is a big one, which applies to me certainly, and maybe some of you. Genetics, hormone levels, men and women are different, nutrition, and then the history of prior infections and vaccinations is, of course, very important, what we've had. So I'll stop at this point and see if there are any questions for clarification. This was the part about the immune system before we move on to viruses. I don't see anything in the chat.
Is there any benefit that carries over from having influenza vaccines year after year after year?
We'll come to why that's necessary. It's because the virus changes. It's not purely that you need a boost every year. It's that actually the virus is evolving and your response no longer matches very well. Okay. Well, we can come back to like more questions later, unless someone. All right. Well, I guess I'll, oh, wait. Are IgE blood tests the best way to check immune responses? IgE, if I understand what you mean, is the antibody responsible for allergy. So no, that would not be the best way to check for any other type of a response. But I can show you a bit about assays later if you want, but that would be a particular class of antibody that has a very specialized function. Okay. Well, now, oh, there's a question. Well, side effects of COVID vaccines. We can discuss that in the question and answer period if you'd like, certainly. Okay. So now moving on to viruses and how they change. I'll discuss virus infection in general, then how viruses change with time, and virus host range. And there's a very wide variety of viruses out there. So viruses can infect different sites in the body, the respiratory tract, the digestive tract, skin, the eye, the urogenital tract, and many other sites. They can be transmitted in a variety of ways. Direct contact, for example, shaking hands. Indirect contact would be touching something like a doorknob others have touched. Insect vectors like mosquitoes can transmit viruses, contaminated food or water, and then two very important routes that are not shown in that cartoon, airborne and sexual transmission. Now, some virus infections we can have, oh wait, there's a question now. If flu changes every year, but if you get a shot each year, does that protect from a future flu? If you get a shot for type X, but that variety shows up again, yes, if you would have at least some. When we come to pandemics, you'll see that the 1918 strain protected partially against the 2009 strain because they were related. So that's a good point. Okay. Now, some virus infections we can have only once in a lifetime, and others we can have repeatedly. So why is that? And here are some of the factors. It's not just one. Some viruses change very little over time, like measles, so they're still recognized by previous immune responses and you don't get it again. Some viruses evolve quickly, as we've mentioned, flu and COVID. Some immune responses were not strong enough in the first place and they don't last long enough. And then some viruses remain in the body, although inactive. That's called latent, and they can reactivate. For example, chickenpox virus lurks in the body and can reemerge many years later as shingles. The virus genome is the genetic material that's copied when a virus replicates. It can be RNA or DNA, and mutations are errors in replicating. Some viruses make more than others, and those modified viruses then can be selected if some of them are more fit. So the genome can be one continuous piece. Here's the rabies virus, it's just one piece. Or there can be multiple segments. Here's influenza virus with a bunch of different segments. And that's important because with a segmented genome, it's an extra way for flu virus to change very quickly. It can swap whole segments with some other flu virus. Viruses evolve at different rates. Some are very stable, some are intermediate, and some change very frequently. So that affects how long a vaccine will still match. And a virus can escape when variants emerge that have changed structure.
No.
Is there a question? No, okay. So here are the rates of change for some in the outer, these are the outer proteins. COVID mutates very quickly, flu quickly, RSV is intermediate, and measles, mumps, and hepatitis B are very slow, so they're more stable. Virus host range is extremely important. Some viruses infect a wide variety of animal species, and some very few or only one. Smallpox is the only virus that's been eradicated worldwide. And the reason that was even possible is that there is no animal reservoir. Polio and measles have no natural animal reservoir. Certain non-human primates can become infected from humans, but there's no continuing spread in the wild. So there have been efforts to eradicate polio and measles viruses, but the efforts are incomplete and still perhaps can be pursued. So I'll pause there and see if there are any questions on this part. Do we know why? There's a question, do we know why some viruses mutate quickly and others don't? Their polymerases, the enzyme that replicates their genome, are different in structure, and some make more errors than others. Also, some viruses have a proofreading mechanism that corrects errors, and some don't. So it's in the enzymology of how they do this. Let's see if there are any other. Okay. So we'll go on to the case studies, and there can always be more questions at the end. Respiratory infections are, of course, a big worldwide public health problem. And I'll use as two examples, influenza and COVID. We're quite familiar with them, but probably we've all had one or the other. So here's seasonal influenza. And we're looking at virus being detected and illnesses over a period of many years. And you can see it's an annual peak in the winter, and the names are different. And that represents that the viruses are different. It's not the same virus every year. The structure of some of the proteins has changed. These names represent where the strain was isolated. Oh, and you may notice that many of these are in Asia. There tends to be a directionality, and that helps predict because they may be seen there before they're seen here. So here's the virus. It has a segmented genome, as I mentioned, so it can swap the whole segment. And on the outside, shown in red, is the hemagglutinin, and in blue is the neuraminidase. Those are the targets of the current vaccines, and they evolve quickly. That's the basis of the HN nomenclature. You may have heard of in the newspaper if what's going around is H1N1 or H3N2 or something, or as the composition of a vaccine. That's what it stands for. There's also influenza B, which doesn't use this nomenclature. Then there are other proteins, internal proteins of flu virus that change much more slowly. So it could possibly be future vaccine targets that wouldn't need to be updated as often. Influenza A pandemics are worldwide infection of people by new influenza A subtypes to which they are not immune. And 1918 was the worst. H1N1 emerged. Here's a photo. The hospitals were so overwhelmed that these are temporary beds in an exhibit hall. People could not even be accommodated. And more Americans died from flu during World War I than in battle. Then a pandemic in 1957 switched to H2N2. 1968 switched just the H to H3N2. And in 2009, a different H1N1 came back, but that is what someone asked about earlier. And yes, elderly people who had lived through the 1918 flu did have partial protection against 2009. So it's sometimes a change in the H, sometimes the N, and less immunity. And now I see there are some more questions in the chat, so I'm going to go take a look. Can tell by looking at a given virus whether it's the kind that will mutate. No, that's determined experimentally. Maybe someday they'd be able to look at the three-dimensional structure of the polymerase, but not generally. And then what is the latest information on the source of COVID, animal or laboratory generated? There is no new information that truly resolves that. That remains somewhat controversial. Okay. So influenza has a wide host range in many kinds of animals. So unlike smallpox, it cannot be eradicated. Especially wild birds are a big animal reservoir. That's where a lot of the strains originate. Then you may have heard about H5N1. That's an avian origin virus in birds, including domestic poultry. Then recently, a different H5N1 in cows, and occasionally it spills over into people. And last week, there was an H5N1 infection in one person for the first time. So the flu cannot be eradicated. The currently approved vaccines are strain matched. So the strain to be used is chosen based on worldwide surveillance data, for example, from Asia, and predictions as to what will circulate in the coming season. Then, once the strains are chosen, it's now trivalent, an H1 and H3 and a flu B. It takes months to manufacture it and distribute it. And the protection can wane by the end of a season. So we're at risk if there's a pandemic or if an unexpected strain emerges because the prediction was off. Turning now to COVID-19. SARS-CoV-2 is the name of the coronavirus that causes COVID-19. The genome is all in one piece, and on the outside of the virus is the spike protein. That's the current vaccine target. And let's see, there's a question in the chat. I think I'll save that question. Ask that again during the Q&A session about long COVID. The COVID-19 virus also infects a wide range of animals, so it cannot be eradicated. The vaccines target the spike protein, as I mentioned, and that evolves rapidly, generating variants that can at least partially escape immunity. So the vaccines are updated to improve protection. Like flu vaccines, that's based on strains that have been seen in surveillance. But it's still valuable even if it's not a perfect match. I want to point out that suppose the vaccine strain is chosen and manufactured, but by the time it's available to you, it's not quite matched because the virus has continued evolving. Well, the similarity is still sufficient that there's usually still some benefit. I see there's a long question in the chat that I'll take a look at shortly. Okay. Timing of events can be very important. Viruses differ in how quickly they replicate, how quickly they cause symptoms, and how much virus is needed to infect a new host. So unfortunately in COVID, the virus is replicating and being shed, and it's highly transmissible before a person has any symptoms at all. So they don't know they're infected and they don't reduce contacts yet. Now, how long does the antibody response last? These are people who got a COVID vaccine, and we're looking at their neutralizing antibody response. Higher is more, and each dot is a person. 2 weeks after their first dose, most people have responded. It's kind of variable. 6 months after the first dose, the response has waned, fallen down. If they get a booster 2 weeks later, the response shoots back up. But what about the cross-reactivity on other strains? How broad is the response? Now, this is the same study, and I've just put in the rest of the data on some other strains. So now we're looking at the strain that matches the vaccine and some strains that do not match the vaccine. So 2 weeks after the first dose, the response is pretty good on the original strain, but pretty bad on the other strains. After 6 months, it's pretty low on all of them. If they get a booster, 2 weeks later, the response has gone up on the original strain, but it has also gone up somewhat on the other strains. So the response to the boost is both higher, more uniform, and broader. Prior immunity can't always prevent infection, but if infection does occur, disease is often less severe. And this is especially true with COVID. It can't be completely prevented. These are patients admitted to a hospital, and all of them tested positive for COVID-19. And what we're looking at is the probability of death in the 30 days. It was a look-back study. So if the patients had not been vaccinated, a lot more of them died than in the fully vaccinated patients. So the disease was more severe in the unvaccinated. Now, this is not, some people might feel like, well, it's a vaccine failure. They became infected and were significantly sick, but they were still better off than if they hadn't been. So turning back to the main points, as we know, infectious diseases caused by viruses are a major public health problem. Multiple types of immune responses result from either infection or vaccination and can play roles in protection. Immunity can prevent illness or at least make it less severe when infection does occur. And some viruses evolve over time and escape our immunity, calling for a booster or a new vaccine. And now I'm going to look at this question. Thank you. And now I'll be taking questions. There's an email address there if you think of questions after this session, but I'm going to start by going back to the chat because there are some questions I missed there.